students here, as far as this important term recombinant DNA technology is concerned, these are the two major or important criteria that which you need to be understood with. That is nothing but here the tools of recombinant DNA technology and processes of recombinant DNA technology. And later on the separate point will be there on applications. Okay, so what are the uses and applications in agriculture and in medical field? So yesterday we have seen that how can we isolate the gene of interest. Suppose the gene of interest is found here in this region and we need to isolate that gene of interest. If suppose this is the gene, or what we can say, the gene of an interest which is found in the Bacillus thuringiensis, and we need to isolate it because this whole means this is the complete, the single, what we can say, the DNA piece or DNA strand, the single circular DNA strand is there, and out of this, or we can say here the double circular DNA strand. So out of this, what we need to understand here with is that the first step comes in the recombinant DNA technology is the isolation of the DNA strand. Okay, so in order to the separate or isolate or cut this DNA, we need to have a specific tool for the recombinant DNA technology or for this whole process. Okay, so here we require the scissor, but this scissor is called as the molecular scissor, and this molecular scissor is in the form of an enzyme, and this is nothing but the restriction endonuclease. This restriction endonuclease, it is very specific in its functioning. That endonuclease, nucleus because it cuts the DNA, that's why nuclease, nucleus. Okay, so it enters into this, it cuts the inner part of the nucleus, that's why it is called as nucleic acid, means whether it is the deoxyribose nucleic acid or ribonucleic acid. So endonuclease means it cuts the part which we require or which we need. Okay, now there are different kinds of recombinant restriction endonucleases are there. So these restriction endonucleases cuts the specific part of DNA. The specific restriction endonuclease cuts the specific DNA segment. Okay, so they are very restricted in its cutting, or what we can say in the region of its cutting. So whatever the region or nucleotide sequence we have here, so this specifics means out of this whole nucleotide sequence, if there is a sequence like what we can say the guanine, adenine, thymine and cytosine. Okay, this is six sequence will be cut by this restriction endonuclease, namely as